All right, we're continuing on with lecture 2, how we get our body parts. This is 2C, female development. So what happens to form the typical female reproductive system? So typically female chromosomes are XX. You'll remember that that X chromosome is necessary for life. You cannot form a human being without it, but there aren't really any sex-specific or reproductive system-specific genes on it for the most part, but we still call it a sex chromosome. That's all right. So what's going to happen in typical female development is that there's no SRY gene present because that's normally only on a Y chromosome. So they'll often talk about how the default pathway is female. So what that means is in the absence of a signal to do otherwise, the embryo will develop as female. Default seems a little derogatory, so I almost want to say the normal pathway, but that's semantics. Okay, so the gonads are going to wait around. So if the SRY gene is present, it usually sends the signal at about 7 weeks. You might recall from male development. They're going to wait around an extra couple weeks, make sure it's just not late to the party. And if they don't get a signal, then they're going to go ahead and become ovaries. So once the gonads become ovaries, where's my animation? There we go. So once they become ovaries, they're not really going to make any hormones, right? So ovarian hormones are things like estrogen and progesterone. We're inside a pregnant woman. There's so much estrogen and progesterone around, like the fetus doesn't need to make any. That's just a wash in it. So what the embryonic ovaries are going to do instead is start making eggs. So the babyest egg cells are called oogonia. Oogonia, strange word, and then they turn into oocytes. And by the time a female-bodied baby is born, they have about 500,000 oocytes in their ovaries. That's all they're ever going to have. You stop making new ones once you're born. So you just start with all the ones you're going to have. So the ovaries are very, very busy making oocytes. They are not making hormones in any significant way during fetal and embryonic development. So internally, since the ovaries aren't making MIS, right? Normally the Sertoli cells in the testes make MIS, not any of the cells in the ovaries. So in the absence of MIS, those Mullerian ducts, those pink tubes, are going to fuse together at the bottom and form the upper part of the vagina, the uterus, and the fallopian tubes out to the sides. Right? So that's pretty cool. So the upper part of the vagina, the uterus, and the fallopian tubes are going to get formed out of those Mullerian ducts. We also don't have any testosterone to speak of because the ovaries aren't making it, right? Those are normally made by Leydig cells in the testes. So without testosterone around, those Wolffian ducts, the blue ones, just disintegrate and go away. So we're left with your typical internal female reproductive system. The other thing that's going to happen is we need to form the rest of the vagina. So the rest of the vagina is going to form, so that lower 2/3 is going to form from that urogenital sinus, which you can kind of see on both of these diagrams here. Not the best resolution, I know. It's a hard diagram to find. Okay, so the vagina is a really interesting structure because the lower part of it, the lower 2/3, form from the urogenital sinus, which otherwise forms the urinary system, the urethra, the tube you pee out of, the urinary bladder. But the upper part of the vagina is formed from the Mullerian ducts. It's kind of neat. It's like a hybrid, a chimera. Externally, because we don't have testosterone around, again, the default pathway is that external genitalia is going to feminize. So what happens is that the genital tubercle becomes the clitoris, right? Which is, in case you haven't seen, so the clitoris is this little nubbin that you see here, although there's more to it than that, as we'll talk about in a couple of weeks. And then the genital folds are going to form the labia minora, those inner smaller lips of the vulva. The labioscrotal swelling is going to form the labia majora. And of course, that urogenital sinus is going to form, let me see if I can use a different color here. It's going to form the urethra here in That's that little tube that you pee out of, and the lower part of the vagina here. Okay, so in the absence of testosterone, the external genitalia is going to feminize. So internal development depends on the presence or absence of both MIS and testosterone. So as we can see kind of on the left side of this slide, that if the SRY gene is present, you make the gonads into testes, you make MIS and testosterone, then the Müllerian ducts degenerate and the Wolffian ducts persist. If the SRY gene is not present, and then you don't have testes, you have ovaries, you don't have MIS, you don't have testosterone, then the Wolffian ducts disintegrate and the Müllerian ducts persist. When it comes to the external genitalia, it's all about whether testosterone is present or not, right? So the external genitalia, you only need to know about one hormone, and that's testosterone. If it's present, you're going to masculinize. If it's not, you're going to feminize. So one of the things that we talk about in biology is homologs, right? So homo means same. Logos means like tongue as in native tongue, as in where you come from originally. Or it can also be used as in like logos meaning wisdom as in logic, right? So knowing where you come from. So if you look, I know this is a busy table, you don't need to memorize it, but I want you to see these relationships. So if you look in the middle, you see that indifferent structure, right? So you start off with an undifferentiated indifferent gonad, and then you can see you have two different pathways. If the SRY gene is present, well, then you're going to turn into testes. If the SRY gene is not present, you're going to turn into ovaries. All right, so you can see how each of those initial structures, most of them have a homolog in the opposite sex, right? With the exception of Wolffian ducts or Müllerian ducts, because you end up with one or the other, not both. So the genital tubercle, right, forms the clitoris in female-bodied people and much of the penis in male-bodied people. So if you hear somebody say something like, well, the clitoris is really just a small penis, they're right, right? Or you could say the penis is just an overly large clitoris, right? Either way, but they are homologous structures, right? The underside of the penis forms from those genital folds, so that is a homolog of the labia minora, the smaller lips on the vulva. And so it's just kind of really neat to see, and the labia majora are homologs with the scrotum. So kind of cool stuff. And I know my video feed of my talking head is in the way of this entire table, so I'm going to go to the next slide and make sure I delete the video from that so that if you want to pause it and take notes, you can. Here it goes. All right, so in summary, typical female development is that you have XX chromosomes, therefore no Y chromosome, therefore no SRY gene. With no SRY gene, the gonads are going to wait around without a signal to do otherwise. They're going to become ovaries. They're not going to make any MIS, and they're not going to make any testosterone. So with no MIS, the Müllerian ducts are going to persist and turn into the upper vagina, uterus, and fallopian tubes. With no testosterone, the Wolffian ducts are going to go away, and the external genitalia is going to feminize and become a vulva. We also then talked about homologs, which are those parts that develop from the same starting structure, which is just kind of a cool thing to think about. All right, so our next part of the lecture is going to be 2D. We're going to talk about differences, because Mother Nature is not merely just this boring. There are lots of different combinations that we can get when it comes to sexual development.